Hey, you look awesome. Which reminds me, let's talk about the autoimmune response.
That sounds like a great freaking idea.
I thought so too. You're awesome and smart.
The human immune system has 3 main lines of defense against foreign microbes. Did you know?
I did not know that. What are they?
The first line of defense are the skin and mucous membranes. Second, there is a protein counterattack which consists of cellular and chemical defenses to non-specific pathogens.
Wow. This is starting to sound complicated.
Stick with me, there is one more. It is a specific immunity which consists of a cellular attack and antibody production against specific pathogens. Our first line of defense, our skin, is our largest organ and makes up 15% of our total body weight.
Wow.
Wow indeed. Our skin has 2 distinct layers, the outer epidermis and the inner dermis. A subcutaneous layer of fat lies underneath the dermis and is called the hypodermis.
This sounds gross.
This is actually quite a beautiful thing. The fat-rich cells of the hypodermis act as shock absorbers and insulators. And get this, microbial growth is inhibited by oil and sweat glands on the surface of the skin, a result of the acid produced. Specifically, our sweat contains the enzyme lysozyme, which attacks and digests bacterial cell walls.
I sure am glad the enzyme does that, because I would not want to.
Well, in a way, you are. The lysozyme in the digestive tract also breaks down bacterial cell walls, and the acidic environment of the stomach kills microbes as well.
Does our respiratory system factor into this?
Yes. I am glad you asked. In the respiratory tract, cells of the bronchi and bronchioles secrete mucus which trap microorganisms. The cells also possess cilia which sweep the mucus toward the glottis.
You were sounding more like a doctor than before.
Well, this is biology, after all. Now, a quick word about specific and non-specific defenses. Let's start with non-specific defenses first.
Sounds great to me. Do you have anything to drink?
No. There are 3 types of white blood cells that roam about the bloodstream, much like policemen who are looking for you, my friend. These are called macrophages, neutrophils, and natural killer cells. All 3 of these can tell if a cell is foreign, much like how I can tell that you are different from myself and that you are a foreigner. When a blood cell fails to make this distinction, the result is an autoimmune disease.
Yikes.
Yeah. Macrophages patrol the blood as precursor cells called monocytes and will also ingest bacteria. Neutrophils also ingest bacteria, but also release chemicals which kill all cells in a given area. The natural killer cells will attack cells that are infected by a virus, as well as cancer cells. They will actually puncture the membranes. Penetration of the skin leads to an inflammatory response. This can be divided into 3 stages. The first is the chemical release of histamine and prostaglandins. This leads to the second stage, the expansion of blood vessels, which allows blood to flow more freely and the vessels to become more permeable. Redness and swelling typically accompany these activities. Thirdly, phagocytes migrate to the site of the infection or injury. First are neutrophils, and then monocytes or macrophages.
Pretty tense stuff.
Surely it is, my brother. Surely it is. Now get this, when macrophages attack, they send a signal to your brain that says, raise the roof. I mean, raise the temperature.
But would it not cause a fever?
Precisely. Fevers inhibit microbial growth, bro. It is a delicate process, however, because high temperatures of 105 degrees Fahrenheit are often fatal.
Scary.
A final word on the non-specific defenses of the body. There is a complement system in the body of 20 different proteins. They circulate freely in blood plasma in an inactive form. They will aggregate to form a membrane attack if they encounter any soldiers, tanks, or airplanes.
What did you just say? Did you say tanks?
Just making sure you were listening. What I meant to say is that they will attack bacterial or fungal cell walls. In the process, they form holes in the invading microbe, so water can rush in and explode the microbe open.
Could you say that this system basically augments the effects of the body's other defenses?
Precisely. Now, on to some specific defenses.
Sounds great.
Specific immune responses target specific bacteria or viruses. These white blood cells ignore everything but the exact thing they are going after.
It would be nice if our body had a way to distinguish between itself and foreign cells.
There is. All cells have protein markers on the surface of their walls or membranes. Human body cells have something called MHC proteins, or major histocompatibility, on their surface. Cells that are foreign to the body do not have these MHC markers, which are unique to each individual. Instead, they have other proteins which are considered to be antigens.
What is an antigen?
Just like a woman, it is complex.
Hectares, ha ha. Funny.
Seriously, it is a complex molecule, such as a protein, that evokes a specific immune response because it is foreign to the body. There is a white blood cell called a lymphocyte, which is involved in specific immunity. They distinguish between normal MHC body cells and those that have been infected with a foreign antigen.
How are they able to identify the specific pathogens?
By the antigens that are present on its surface, of course. There are two main types of lymphocytes, called T cells and B cells. T cells originate in bone marrow and migrate to thymus. There are 4 types of T cells.
Of course there are.
Helper T, which initiate the response of other lymphocytes. Then there are cytotoxic T, which are lyse-infected body cells. Third are memory T, which provide a quick response on re-exposure. Last, there is suppressor T, which terminate the immune response.
I love tea.
I bet. Next we have B cells. These originate and mature in the bone marrow, and then circulate in the blood and lymph. Upon antigen exposure, they would proliferate into plasma cells or memory cells, either producing antibodies or providing a quick response to re-exposure.
Sounds like they are on the ball. So tell me, what is it exactly that triggers or initiates the immune response?
Macrophages initiated by inspecting the surfaces of the cells they encounter. When they come across invaders with antigen, they ingest them and partially digest them. They then display the antigen on their surfaces as a complex with MHC and are now referred to as antigen-presenting cells.
Pretty alarming event.
Actually, that is exactly what happens next. Once the macrophage becomes an antigen-presenting cell, it secretes an alarm signal, the protein interleukin-1. This protein stimulates helper T cells to bind to the macrophage. T cell receptors can only interact with cells that have the MHC antigen complex.
Well, it seems that macrophages are really important to start the immune response.
Just like remembering your anniversary, the importance cannot be understated. The binding of the T cells to the macrophages causes them to become activated and initiate 2 responses, the cellular immune response, where T cells attack antigen-presenting cells, and the humoral immune response, where B cells release antibodies into the blood that mark pathogens for destruction.
Woohoo! Destruction.
Activated helper T cells also give rise to the memory helper T cells, which respond to secondary infection by the same pathogen. The helper T cells also secrete the interleukin-2, stimulating proliferation of cytotoxic T cells.
Tell me a bit more about the cytotoxic T cells, brother.
Of course. Cytotoxic T cells destroy antigen-presenting cells that display the specific antigen for which they were activated. They puncture holes in the plasma membrane and produce memory T cells. They can also attack cells with foreign or altered MHC, as a result, for example, of transplanted tissue or cancer cells.
Are B cells similar?
Well, B cells process antigen like macrophages. Antigen is taken and presented on its surface as an MHC complex. Helper T cells bind to the antigen-presenting B cells and secrete the interleukin-2 also, stimulating the production of plasma and memory B cells. The plasma cells produce antibodies, proteins that bind to antigen and flag cells for destruction by macrophages, NK cells, or complementary cells.
Again with the destruction.
Memory B cells will circulate the blood and lymph, awaiting a future exposure. The second response is amplified by about 1,000,000-fold.
I wish I had 1,000,000 in my billfold.
I bet you do. Please stay on subject.
Okay. You mentioned proteins earlier, antibodies. Could you elaborate a bit on those?
Surely, my brother. There are 5 different subclasses of antibodies. IgM, which is the first to be secreted during the primary response. Then IgG, the major one secreted during the secondary response. Then there is IgD, a receptor for antigen on the surface of B cells. Also is IgA, the type in external secretions like saliva and milk, and then IgE, which promotes the release of histamine causing allergic reactions.
Pretty cool stuff. This is very interesting.
Well, it is not money, but yes, it is interesting. Anyway, humans only contain a few hundred receptor-encoding genes that can make antibodies. B cells can make an estimated 10 to the 6th power different antibody molecules, up to 10 to the 9th.
What? Oh my! I hate math. How is that even possible?
The immune receptor genes are assembled by a process called somatic rearrangement. DNA segments that code for different receptor parts are basically stitched together. Further antibody diversity is generated by imprecise DNA arrangements and random mutations.
Mutations like the X-Men? Or Wolverine? I love Wolverine.
I will pretend like I did not hear that. Listen to me now. The first encounter with a foreign antigen results in the primary immune response, right?
Yeah.
Well, only a few B/T cells can recognize the antigen at that time. The binding of antigen to its receptor on a lymphocyte starts something called clonal selection, in which clones are produced through cell division. Of course, the second encounter with a foreign antigen is termed the secondary immune response.
Ooh, I know. I bet there is a much larger response this time, right?
Correct. This time, there is a large clone of lymphocytes that can recognize the antigen, and the immune response is now swifter and stronger than before.
This has been some interesting stuff. I sure am glad I do not have a plasma cell sticker on my car, though.
Why is that?
It would say kill me.
Oh, ha ha ha, very funny.
What are you going to do now?
I am going to go ahead and grab myself a drink.
That sounds delicious. I think I will join you.